Hi, everyone. Thank you so much for being here today for this extremely important session. So I'm Jesse Kirkpatrick. I am a PSC researcher, and I am a family member of a PSC patient. And I think we can all agree that there is no one in the world who cares more about the goal of finding a cure for PSC than us here in this community. And for those of you who were at Dr. Feigenbaum's presentation earlier this morning, you'll have a pretty good idea of just how powerful it is when a patient-centered organization takes an active role in defining what are the most important research priorities for an organization, for the patients within it, and to bring together patients, physicians, researchers, pharma, and more toward the common goal of curing a rare disease. So the presentation that you're about to see represents the launch of PSC Partners' effort to do exactly that. But a quick note to our audience. In order for this to work, we, as a community of patients and their loved ones, need to take ownership of this moment and really trust in our ability to make a difference. That means engaging with the process, coming up with ideas and questions, not being shy, contributing your voice. I think Dr. Feigenbaum really said it well this morning when he said that even if you don't have a background in science and research, you know what you worry about. You know what your loved one who suffers from PSC worries about, and you can contribute that voice. And so now I will send it over to Dr. Ruth-Anne Pai, who is PSC Partners' Director of Research Strategy, and who will be unveiling the PSC Partners Research Agenda and kick off the International Collaborative Research Network. Dr. Pai completed her PhD in immunology with Dr. Feigenbaum at the University of Pennsylvania and has spent the past year leading our team at PSC Partners in our new laser-focused effort to accelerate treatments and a cure for PSC. So Dr. Pai, please take it away.
Hello, everyone. I'm Ruth-Anne Pai, the Director of Research Strategy at PSC Partners Seeking a Cure. I joined PSC Partners last April after completing my PhD in immunology at the University of Pennsylvania. I completed my thesis work in Dr. David Feigenbaum's lab, practicing the collaborative network approach that David shared during his keynote earlier today. Now, over the past year at PSC Partners, I have been so honored to get to know many of the incredible patients, caregivers, researchers, and clinicians who are part of the PSC community. Today is a very exciting day because it marks the official launch of the PSC Partners International Collaborative Research Network, the ICRNetwork. During this presentation, I'll share what the ICRNetwork is. And by being actively responsive, you are already an important part of this major effort. Then I'll share a key goal of the ICRNetwork this year, the discussion and prioritization of a PSC research agenda, and ultimately a 5-year strategic research plan for PSC Partners, where we as a community will be a small but mighty force towards treatments and a cure for PSC. So first, what exactly is the PSC Partners ICRNetwork and how will it function? The ICRNetwork is PSC Partners' name for our collaborative network model, first defined by the Castleman Disease Collaborative Network, the CDCN, in 2016, and described by Dr. Feigenbaum in today's keynote presentation. The model is a formal connection of key stakeholders with the same goal and core values, identifying treatments and a cure for rare diseases. The important pre-work to this process is the identification of outstanding patient needs, which we've been collecting over the past 2 years through the Roadmap Initiative and the Patient-Focused Drug Development Forum, the PFDD. Now, PSC Partners is prepared to work with key stakeholders, including researchers, clinicians, patients, industry, and regulators in order to perform the following steps. To identify the partners best able to address the research question. Then to coordinate efforts to raise funds and build additional resources needed for the research project. To support patient recruitment and the collection of research samples. To collaborate with researchers as they perform experiments and analyze data. To partner with researchers to translate research findings and share them directly with PSC patients and caregivers. And then to continue this cycle to again prioritize outstanding research questions that address PSC patient needs. During my time at Penn, I saw firsthand how this patient-centered research approach can accelerate research as it led to the identification of 2 new treatments for Castleman disease. Now, I'm very hopeful that by adopting this approach, we can accelerate the pace and success of PSC research by working together as a united ICR network. As you can see, there's a place for everyone here today to support these efforts. In the spirit of this model, PSC Partners now invites all stakeholders, patients and caregivers, researchers, clinicians, and industry partners to unite efforts through PSC Partners and the ICR network. Through our work together today and over the next few months, we aim to prioritize just a few research questions to pursue in research projects over the next 5 years. As we all know, there is no drug approved by the US FDA or the European EMA to treat PSC. Through the 2020 PFDD meeting and the Our Voices survey, our patient community clearly prioritized the development of drugs to slow or stop the progression of PSC. It's important that our research agenda include the patient voice and also consider patient-focused drug development as a means to reach a world where a PSC diagnosis comes with a cure. While today marks the launch of the ICR network, our work together really began months ago when PSC patients and caregivers shared questions to guide research and drug development during the 10 Roadmap Initiative meetings and through the externally led PFDD meeting for PSC in October 2020. This work would not have been possible without the dedication of the entire team at PSC Partners, our amazing patient and caregiver volunteers, and dozens of researchers and clinicians who have volunteered their time to facilitate these conversations. In a way, the ICR network formed last year through the passion and care of our diverse and broad PSC community. These conversations guided the selection of key questions that PSC patients ask throughout their journey with PSC. Then, following a deep review of hundreds of scientific articles, Dr. Jesse Kirkpatrick, a PhD scientist, family member of a PSC patient, and a PSC Partners board member, joined me in developing the very first draft of a PSC Partners research agenda. We also met virtually one by one with 8 international PSC experts to discuss areas of research ranging from the immune system and the environment to fibrosis and biomarkers. We incorporated the expert opinion of these PSC researchers and organized their research questions across each of the key questions asked by PSC patients along their journey with PSC. Today, we are very excited to share with you all the very first draft of the PSC Partners Research Agenda. As we review the agenda, I invite you all to think deeply about which of these questions we should discuss in more depth and pursue over the next 5 years. Please enter your thoughts into the chat and share your questions in the Q&A box. In just a bit, we will have time for a panel discussion and we'll hear more from these PSC expert researchers. Through our continued work together during and after the sessions, we will narrow our focus and prioritize the PSC patient voice. While the agenda will look quite expansive and ambitious today, we will narrow our focus significantly to address the most important questions and develop a strategic plan to accomplish our goals over the next 5 years. Now, let's unveil the draft PSC Partners Research Agenda. As I mentioned earlier, the PSC patient voice was centered in this work and guided the selection of key questions shown here that PSC patients ask along their journey. While these questions don't reflect the full spectrum and many diverse journeys of PSC patients, they highlight our overarching needs and the questions that keep us all, patient or researcher, caregiver or clinician, up at night. First, what is the initial cause of PSC? What causes PSC to progress? How do we detect PSC early, monitor it effectively, and predict long-term prognosis? How do we improve and accelerate the development of new treatments for PSC? How do we improve transplantation rates and success? How do we prevent recurrence? And how do we prevent the development and progression of cholangiocarcinoma in PSC? These questions are not new. In fact, hundreds of research studies have been performed over the past few decades, uncovering important aspects of PSC and leading to countless new questions as well. In this way, scientific research builds on previous findings. More so today than ever, we are so well positioned to make progress because of the dedication and strength of PSC research groups and collaborations. While the information in the draft agenda is complex and full of scientific terms, PSC Partners will always be here to answer questions and translate research findings. Now, I'll walk us through each section of the draft agenda, sharing key research questions on each slide. The text on the slides will include the questions from the draft research agenda in scientific language, and what you hear from me may not align well with what you see on each slide. Rather, I'll do my best to present the questions using lay language, simplifying the text and centering on why these research questions are so important. First, when PSC is diagnosed, we may ask, what caused this disease? In other words, what triggered my PSC or caused me to have PSC in the first place? We don't know yet what causes PSC, but many factors are suspected to play a role, including genetics, the environment, the immune system, such as autoimmune disease, and factors in the gut, such as IBD. Understanding the initial cause of PSC may help us in a number of ways, such as in the identification of biomarkers, like a simple blood test, that can be used to better screen for and diagnose PSC. The development of new and better models to study PSC, and the development of strategies to treat PSC earlier and more effectively. Next, many will ask, what will cause my PSC to get worse? Is it my IBD or my immune system? What is fibrosis and why is this scarring happening in my liver? Can I do anything to slow progression? Is there something in my environment, like my diet, that I can change? These are all questions that guide research and lead to countless avenues to better understand PSC and to identify new treatments. For this reason, it's important to further organize our questions into the following on the slide. What is the link between the gut and liver in PSC? What is the nature of the inflammation in PSC? What is the nature of fibrosis in PSC? And finally, how do genes and the environment contribute to PSC progression? Now let's dive into these 4 areas around PSC progression, one at a time. First, many PSCers ask about the relationship between their PSC and IBD. And this is a very important area of investigation for researchers as well. We can simplify the complicated questions on this slide in this way. When the gut is altered, like in IBD, when there is inflammation, how does that relate to inflammation in my liver? Do my PSC and IBD go hand in hand, or are they separate diseases that can occur at the same time? Do I, a PSC patient with IBD, have the same type of IBD as those who don't have PSC? And should my doctors treat me the same way? And does my PSC change the severity of IBD that I have? We know that as many as 80% of PSC patients also have IBD, and yet not much is known about how IBD and changes in the gut impact the development and progression of PSC. So far, we've looked at just the tip of the iceberg, and PSC Partners will be working towards sparking and guiding collaborations between PSC and IBD researchers to uncover the mystery of what's called the gut-liver axis in PSC. As I mentioned earlier, the most important question selected by PSC patients through the PFDD meeting is to identify treatments that slow or stop the progression of PSC. And for each of these areas of progression, we must consider research questions to address this need. As I walk through these 4 sections, you'll notice questions highlighted in purple, which are important to address in order to find new treatments to slow or even stop the progression of PSC. When it comes to the gut-liver connection in PSC, we can ask how best to modulate this axis to stop the progression of PSC, perhaps through diet, fecal transplant, or supplementation, such as substances tested in preliminary studies like curcumin and omega-3 fatty acids. The more we learn about the connection between IBD and PSC, the better positioned we'll be to identify effective treatments. Next, what is the nature of the inflammation in PSC? Many PSC patients and researchers ask the same question: how does inflammation contribute to PSC? Inflammation is caused by our immune system, a complex network of cells and proteins contributing to an orchestrated immune response. Inflammation is beneficial in helping us get over infections, but in PSC, the immune response is inappropriate and leads to inflammation of the bile duct and also to fibrosis, scarring of the bile duct. Not much is known about the early and late stages of inflammation in PSC, and in particular, immune cells and their roles in the liver. Almost all of the researchers that we spoke with highlighted how important it is to characterize immune responses in the liver and in the gut, both in early, late, and middle stage PSC. Shown here are just 3 of the questions proposed by immunologists investigating PSC. Now, many PSC patients have experienced firsthand that drugs targeting the immune system, including many used to treat IBD, do not seem to affect the progression of PSC for many. Researchers think this may be because we are studying PSC too late in the disease progression, when these early immune responses may no longer be present. And so we may be trying the wrong drugs at the wrong time. Our question here in purple addresses this need. By understanding the full spectrum of immune response from early to late PSC stages, our hope is that we may find new strategies and the ideal timing to target both early and late inflammatory immune responses. Next, I mentioned that the immune system contributes to fibrosis, scarring of the bile duct in PSC. But in fact, there are many complex processes that may drive fibrosis, and the questions here require a deep understanding of cell biology. Within our liver and all the way down to our bile duct, cells communicate with one another in ways that may lead to progression of fibrosis. The exact processes and mechanisms driving changes in the liver in PSC and leading to fibrosis still require investigation. Fortunately, there are already many brilliant researchers dedicating their time to addressing this key research area. And if we aren't able to prevent the initial development of liver scarring early in PSC progression, the next best thing may be to target and stop fibrosis in its tracks. As we tease apart cellular mechanisms like genes, proteins, and cellular responses that contribute to the liver scarring, we can identify precision treatment strategies targeting those mechanisms. There are some very exciting strategies that are picking up steam right now, including gene silencing, nanoparticle delivery of treatments to the liver, and regenerative medicine, just to name a few. Our 4th and final topic under this question of what contributes to the progression of PSC is focused on genes and the environment. We already know that PSC patients have certain genetic differences from people without PSC, but we don't really know why those differences lead to PSC. The research questions on this slide boil down to 2 important questions. First, could our genetic background impact the way that our immune system, gut, and liver function in the context of PSC? And could various aspects of our environment, like our diet and chemical exposures, impact the progression of PSC? By studying the role of these different genetic and environmental factors, we may learn more about what causes PSC to progress. These studies may also help us predict which patients are more or less likely to progress. As we search for new PSC treatments, the questions here in purple are very important to ask. While we cannot change the makeup of our genes, we may find ways to target the downstream consequences of our genetic background. Similarly, we may find ways to modify our environment, such as through our diet, to slow PSC progression. Identifying and studying these genetic risk factors and environmental exposures may lead to new PSC treatments and also impact the ways in which we detect, monitor, and treat PSC, which will be our next topic. At the end of the day, each of us here wants to improve quality of life and outcomes for all PSC patients in our community. With all of the research described above and the identification of new potential treatment targets, we next need to direct our efforts towards two crucial areas: drug development and improved clinical care. Let's begin with clinical care. Now, having asked what causes PSC and what contributes to its progression, many PSCers ask questions like, what will my next 5 or 10 years look like? How quickly will my PSC progress? How can we better monitor my PSC? One way to answer these questions is to identify biomarkers and screening tools to monitor PSC and predict prognosis. A biomarker is a biological molecule found in blood or other body fluids like urine or tissues that is a sign of a condition or disease. For PSC, it's important for these biomarkers to be non-invasive, meaning they're easy to collect and lead to minimal discomfort or complications. Our hope is that we will find biomarkers in blood, stool, saliva, or urine that can be measured easily at low cost and over time to aid in PSC diagnosis, management, and treatment decisions. Now, clinicians may be familiar with these common questions. What other treatments can we try to stop my PSC? And when will a cure be available to me? As we strive to improve clinical care and treatment of PSC, we first need to rigorously study promising treatments in clinical trials. This process of drug development is so critical and involves everyone here today. Through the multiple phases of drug development, novel treatments are studied in clinical trials to determine whether they are safe and effective in treating PSC. In order for these trials to be successful, we must work together to ensure we capture the most important clinical changes, including patient-reported changes in PSC symptom frequency and burden. As we begin our 5-year strategic research plan, PSC Partners has already prioritized the development of a PSC-specific patient-reported outcome measure, or PROM, that is designed specifically for clinical trials. We hope you'll join us tomorrow at 12:45 p.m. Mountain Time to hear more about this project from the academic research leads, Dr. Yvonne and Dr. Reeve. In addition, clinical trials in PSC are often long, and we ask a lot of PSC patients enrolled in these trials. As a result, we as a community should prioritize the identification and study of biomarkers that are able to tell us early and with great accuracy whether a drug is slowing the progression of PSC. PSC Partners and PSC Partners Canada have already begun prioritizing all of the questions on this slide as we strive to improve and accelerate drug development through our work both with drug developers and through the WIND initiative. You'll hear more about this WIND initiative tomorrow during the WIND think tank session. Now, let's take a step back and consider that over the course of the next 5 years, as we work to accelerate new treatments through the drug development pathway, there still remains a great need to improve transplantation rates and success. In the absence of effective drugs, and as PSC progresses, we encounter many questions around transplant waitlist, complications, and access to treatment. And after transplant, many ask what the risk is of developing recurrent PSC and how we can lower this risk. The voice and the needs of the post-transplant community are so important and must also be heard. And last, but certainly not least, many PSCers live in fear of developing cholangiocarcinoma, or CCA. The first 3 research questions on this slide clearly align with those asked most frequently by PSCers. What aspects of PSC contribute to the development of CCA? How can we best treat PSC in order to prevent the development of CCA? And how do we detect CCA while it's still curable? While the risk is low, we all want to know more about how cholangiocarcinoma develops and what we can do to prevent it or diagnose it earlier. Now that we've walked through the draft research agenda, let's take a step back and discuss our next steps for narrowing our focus and discussing these topics together. You can find both the full-length draft research agenda and a streamlined research agenda for patients and caregivers on the PSC Partners ICRNetwork webpage on pscpartners.org. There should be a link to this page in the chat as well. Next, we'll prepare for a live Q&A session with panel discussion in just a few minutes. Please participate in this panel by entering your questions into the Q&A box. Later today, we'll take a deeper dive into this draft research agenda during the think tank session. At 11:20 a.m. Mountain Time, everyone in attendance will have the opportunity to discuss aspects of the research agenda in small groups of 10 to 20 during the very first ICRNetwork think tank. This is an exciting opportunity for all of us to get together and discuss PSC research in a laid-back, fun environment. A PSC Partners moderator will walk us through a list of questions, and you'll have time to share your thoughts, questions, and hopes for future PSC research. We ask everyone in the audience to stay on, listen, or participate because your presence is what will make the research network stronger. Now, I'll finish up with an overview of how we as a community, through the ICRNetwork, will translate our research agenda into a strategic research plan. The draft research agenda presented today is expansive and reflects outstanding research questions highlighted in the past work of hundreds, if not thousands, of researchers. Realistically, we must now work as a community to narrow and focus our efforts as a united research network. Today, and in the coming months, we will prioritize a list of actionable areas to dedicate PSC Partners resources and our community efforts to over the next 5 years. The areas of research that we choose to focus on and that PSC Partners will pursue will reflect just one of countless efforts for the PSC community. And yet we truly believe that we can make a difference as we center the PSC patient voice and needs. Now, the path to success may look a bit different for each research question. For example, we may choose to address research questions through the ongoing PSC Partners grant program, which will continue. Alternatively, PSC Partners may develop a request for proposals, calling researchers to submit their plans for their own research projects addressing a key research question. A third option, PSC Partners may pursue a direct collaboration with researchers, which is what we have chosen as the best avenue towards developing new patient-reported outcome measures for PSC. As part of our strategic research plan, we will also identify tools and resources needed for each research project. These may range from access to technology and patient samples to coordinated fundraising efforts driven by PSC Partners and the patient community. To close, I want to draw your attention to the graphic on the right. Here we see 3 circles with key research questions at the center. I hope that you will leave here today with an appreciation of the important role that we all play in driving forward PSC research. At PSC Partners, we view patients and caregivers as equal partners alongside researchers, clinicians, drug developers, and funding partners. Through open collaboration and sharing of research samples, clinical data, experimental models, and donations to PSC research, we can create a world where a PSC diagnosis comes with a cure. Thank you for being here today. And again, I want to encourage you all, patients, caregivers, researchers, clinicians, and industry, to all join us in the think tank breakout rooms later today. We need all perspectives in order to create the best research agenda possible. Thank you.
Thank you, Dr. Pai. That was incredibly exciting. I was involved, as Dr. Pai mentioned, in coming up with this draft research agenda, but it's just so incredibly exciting to see her present it in front of all of our entire community. And really finally launch this effort. So we will now turn to our panel of experts to discuss both the PSC Partners Research Agenda, the ICRNetwork, and more. And so joining us here on the virtual stage are Dr. Ruth-Anne Pai, who I've already introduced, as well as Dr. Chris Bowlus, Dr. Christoph Schramm, and Dr. Michael Trauner. And so I'm just going to give a quick introduction of each of our panelists. So Dr. Bowlus is the Chief of Gastroenterology and Hepatology at University of California, Davis School of Medicine. And he has performed extensive research on PSC and other autoimmune liver diseases. And in my opinion, one of his most seminal studies that he published was a recent paper that showed that obeticholic acid improved liver function tests in PSC patients. And so this is a really promising therapeutic candidate that's already gone through extensive trials in PBC and is now entering trials for PSC. So thank you, Dr. Bowlus, for that. Dr. Schramm is a senior physician in hepatology and gastroenterology at University Medical Center Hamburg-Eppendorf in Germany. And his clinical and lab research into PSC have really dramatically increased what we know about the role that the immune system plays in PSC, the immune system being one of the research areas that Dr. Pai spoke about in her presentation. And in fact, his lab recently published the first ever single cell RNA sequencing study in PSC. And what that basically means is that he and his team characterized the immune cells that infiltrate the liver in PSC with more depth and detail than ever before. So that was also an incredibly exciting paper to read. And then Dr. Trauner is the chair of gastroenterology and hepatology at the Medical University of Vienna. And Dr. Trauner has made groundbreaking contributions to another area that Dr. Pai spoke about, which is bile acids and the role that bile acids play in PSC, and has played a very pivotal role in the development and validation of nor-ursodeoxycholic acid, which is now referred to as noracolic acid in PSC patients. And that's another very promising therapeutic candidate for patients with our disease. So we have an incredible group of people here today who have devoted their careers to PSC. I'm so grateful to all of you for being here. Thank you so much. And finally, once again, I'd like to call on our audience. There are many of you here today. You have combined much more experience with this disease than anyone here on this stage. So please trust your voice, trust your experience, and share any questions or ideas that you have using the Q&A feature. And you can also write them down, bring them to your think tank breakout session later today. And even if we don't get the chance to get to all of the questions, know that we see them, we're writing them down, and we're incorporating your feedback. And just to remind you that this is our agenda, so it is so important for all of our voices to be heard and incorporated into it. So we have all of our expert panelists here on stage. So I will just start with a general question for all of our panelists. So the research agenda that Dr. Pai has just presented includes 6 key areas. I'm just going to summarize them again, which the key areas are the initial cause of PSC, the factors that lead to PSC progression, the ways in which we can more effectively diagnose and monitor PSC, so biomarkers, drug development, transplantation outcomes, and then the prevention and early detection of cholangiocarcinoma. So I just wanted to go around and just sort of get your first gut reactions to this research agenda, the strengths and maybe weaknesses, and then and what you would add or change to it. So I'll start with Dr. Schramm.
So first of all, thanks for having us during this exciting session, and congratulations to PSC Partners for developing this research agenda. I think it's excellent. It's very comprehensive, as you all saw. I want to just make one point, because Ruth-Anne said that patient's contribution is small. I think from a clinician scientist point of view, patient contribution is really fundamental to make progress, deciphering the mechanisms of disease and what leads to disease progression. And just as an example, if we don't get biopsies from the liver from you, we will not be able to know what is going on in the liver. And using mouse models will not suffice to answer these questions. So thanks to all of you who donate biosamples. I think this is really so important. And my comment to the research agenda is, I don't think we can prioritize one over the other, and some aspects are really overlapping. So once we understand what causes inflammation, we'll probably also understand what causes fibrosis, so scarring in the liver, just as one of the examples.
Thank you. Dr. Bowlus?
Yeah, good morning, everyone. And also thanks for inviting me here today. And I agree with Dr. Schramm said that this is a very comprehensive research agenda. I think that I would just have two brief comments. First, I see in some of the chat, people are asking about patient's voice, or at least one of the comments was, what does that mean? And I think a lot of the comments there kind of bring it up, is that, you know, what is important to patients? What are the things that patients recognize? We as researchers have a certain bias of the way we look at PSC, and we may not focus on certain areas of the disease that are important to patients. There's also a comment about the use of antibiotics as triggers and things like that. And so getting an idea from patients, you know, clues and understanding the disease from their perspective is really key to moving this forward, both in terms of understanding the disease and also what's feasible. We may design the most perfect study from our point of view, but it's not feasible because it's not acceptable to patients, and it's no good. So it takes all of us to really move this field forward. In terms of the agenda, it is comprehensive. I would say my own feeling is that the key here is getting treatments to the patients. While understanding the disease is critical and will provide probably long-term understanding and treatments, targets for treatment, currently we have a roadblock in terms of getting therapies that we potentially could study now, but we don't have the biomarkers or pathway to get them approved. There's just some very practical issues with that process. And so I think some of the plans that PSC Partners and this network have are really going to address that problem we've been really struggling with for the past decade or more. And so to me, that's really the key critical issue to address that this plan is attempting to do.
Thanks, Dr. Bowlus. I appreciate you once again highlighting the role that patients can play. I think it's so important for everyone to hear. Dr. Trauner, your thoughts?
Yeah, thank you very much for having me here. And Ruth-Anne, I really have to congratulate you. That was fantastic. I mean, you basically covered it all. Yeah, and because of that, you have to prioritize. If you really want to achieve a 5 years aim, I think it will be necessary to prioritize and really can emphasize what has been said, that the patient perspective is extremely important, also in terms of defining outcomes. And I think, you know, as a liver doctor, hepatology community, quite honestly, be critical. We have not been doing a good job compared to other disciplines. Hepatology is very hard endpoint, survival driven, you know, things like that. For example, of course, in rheumatology, it's much more important how the patient feels in terms of pain. This is something which may be more difficult with liver disease, but there are many things, how you feel, you know, fatigue, and other issues which are not really well reflected in the endpoints when it comes to clinical study. And I think, you know, if we combine together influencing regulators, how to define endpoints, and again, taking the example of rheumatology, for example, where composite endpoints, where let's say 5 of the endpoints, 2 are hard, biochemical, radiological, and 3 are patient-related outcomes. I think this needs to be integrated much more. And also, you know, in order to achieve those aims, at the end, of course, it comes down to financing. And for example, in Europe, for example, the European program of the European research community is an open call for ideas, where, you know, stakeholders would tell, in your case, the NIH or other fundings agency, what should be the priority. So I think patients should take all the political influence they have, also, you know, in terms of influencing the research agenda of funding authorities, because you even, if you do a great job with fundraising, you will not be able to pay for all of that, what you've listed, which are great aims. I mean, I think one of the things which we may have to change with PSC is to get rid of the S in the PSC, which is the sclerosis, because this is basically something, of course, at the moment how we diagnose the disease, but this is scarring and this is end stage, or let's say very advanced disease. I think we have to move on earlier, to diagnose the disease earlier. And again, here, for example, taking the example of rheumatology, where they start treating patients who had a little swelling of the joint because they think it could develop into rheumatoid arthritis. We have to go back much earlier with PSC, for example, with imaging, where you can image the cholangitis nowadays without the sclerosis. The sclerosis was something, in the cholangiograms, but now with modern imaging, you really can image inflammation. And again, here we have to convince the regulators that this is already the disease, and we only can do that together. So I think early diagnosis and early treatment will be key to really make a turnaround. And it may be even changing the definition of the disease at the end.
Thanks, Dr. Trauner. That's a really interesting point. It's something that you brought up in our discussion about the research agenda back several weeks ago when we spoke. I think it's interesting to think about sort of the different stages of PSC, from the earliest potentially like inflammatory, like inflammation of the liver, towards this more end stage fibrosis. And it is so important for us to really define what those different stages are, and develop biomarkers and potentially treatments for those specific stages. And Dr. Pai, what are your thoughts on this agenda that you very nicely presented?
Thanks, Dr. Kirkpatrick. Yeah, so there definitely is a little bit of a conflict of interest since I was heavily involved in preparing this. But my thoughts are really along the same lines as Dr. Trauner, that we want to consider how we define PSC as well. It's not really captured in the agenda, but as the strategic research plan takes shape and research progresses over the next few years, we might need to reconsider as a field, like Dr. Trauner said, how we define, diagnose, treat, and manage PSC. So perhaps these discussions and shifts in clinical definitions and guidelines will be an area that PSC will focus on down the road in a few years.
Thank you, Dr. Pai. And I also just want to add that if any of the panelists want to kind of like add to or potentially rebut anything that any other panelist suggests, that would be great to have this be kind of an open discussion. So there are so many questions in the Q&A. I will try to get to at least some of them. I also had some questions that I put together for the panel to make sure that we get the very targeted feedback on the agenda. So another question that I had for the panelists, so when thinking about taking this research agenda from what it is now, which is an agenda that, as many of the panelists have mentioned, is extensive and comprehensive, towards something that's actually feasible, into something like a concrete 5-year strategic plan, which areas do you think we as an organization should really be prioritizing, both from the perspective of what is the greatest unmet need, and then also what you think that that's really feasible to tackle? And I'll just go back to in the same order with Dr. Schramm.
I don't know whether it's up to us to advise you what you as patients should prioritize. I think many of the important aspects have been touched upon. Another one is the WIND initiative, which I really like. So to have a natural history cohort of high quality that can be used as a control arm in clinical trials, I think that would be excellent, because it would greatly reduce the numbers of patients we need to include in clinical trials. And I think that will be one of the foci, and I think that's excellent. And I very much like the idea of directly collaborating with experts in the field if you want to speed up the process. If you go via calls and applications, you'll probably lose a year until the work starts. So if you already have done so with the prompts, I would like to encourage you to go on this way.
Great. Thank you. Dr. Bowlus.
Yeah, I would say that one of the considerations you might think about is what could be funded by other institutions versus what really would rely on PSC partners. So I think some of the items for study are really well suited for support through NIH or other funding agencies. But things like the WIND initiative, these natural history studies that are so key, these are not of interest to NIH to fund. So that's where I think, in particular, PSC partners may want to focus some of their resources towards that.
Great, thank you. And Dr. Trauner.
No, I think certainly, developing patient-related outcome measures, I think that certainly, I think that would be a priority also from your side, because this has the most impact on daily life probably. And, a lot of the questions, for example, in the chat were also on diet. And, there are many things which we do not understand at the moment. And let's say there may be single factors, and I think this came out of the chat, which play a role in individual patients. And if you investigate them in a big group, it doesn't have a signal. And this is probably something where artificial intelligence and other things, in terms of data integration may come into play. And also here, I think a lot of, let's say, real life collection of data, be it dietary, monitoring with bio devices, with your apps, in your daily life, which may produce a signal which we do not recognize because it's small and it's subtle, and it may be a signal, as one of the multifactorial hits, and maybe an artificial intelligence. I don't think, artificial intelligence will solve everything in medicine, but it will help to analyze this overwhelming amount of data which are out there. And when we talk about microbiome, and also in the field of IBD, certainly diet, there was a turnaround. For a long time, we told our patients, just eat, what you tolerate and what you think is good for you. And it is Crohn's disease, for example, there's a lot of science now behind of dietary intervention and modulating inflammation. And I think it may be one piece, but it may be an important piece together with others. And this is information which we have to collect. And this is something which you as patients can help enormously. But certainly we need some technical devices which help to collect this information.
Great. Yeah, that's something that I think we haven't really thought too much about in our community. And we have such an abundance of, these like health tracking apps and things that we can really leverage. So it's great to be able to think about that. And Dr. Schramm has his hand raised.
Yeah, just one comment regarding early diagnosis of disease, which is something we as PSC scientists, researchers, can probably not solve on our own, because you need the whole IBD community included. And that may be a topic that PSC Partners wants to tackle, because if you really want to identify patients early on, you would need to screen the IBD patients with whatever method, maybe MRI or with biomarkers, but we likely do not have access to these patient cohorts. So you could facilitate contact between the IBD and the PSC communities research-wise, just as another comment to what to prioritize.
Great. Yeah, that's a great point. Thank you. And Dr. Pai.
These are all great points, great ideas, certainly really helpful feedback. I'll just comment on Christoph's note about the PSC IBD connection and those collaborations. This is exactly what we are looking to do, is to identify researchers to collaborate with across both the PSC and IBD space and ask questions like that. So that's certainly the approach we're looking to take. Thank you.
Great. So because there are 28 questions in Q&A, I've been trying to kind of go through and at least get a general sense of what people are interested in and asking. There are questions here about the use of potentially oral vancomycin. There are questions about why we use biopsies as opposed to other endpoints in clinical trials. questions about environmental exposures and how those could be potentially contributing to PSC. I think that one, maybe just since it is getting quite a lot of attention, maybe if just one of our panelists could comment on the use of oral vancomycin and the strengths of that approach, the potential weaknesses, what might need to be done to really power these types of studies. So yeah, are there any volunteers to tackle the oral vancomycin question?
I'll step into that one if Michael and Christoph aren't willing. Yeah, so the data on oral vancomycin is really intriguing. There is an ongoing study on it. Clearly, there needs to be more done in this area in terms of understanding what are the long-term outcomes of treatment are, what's the safety. There is concern about development of vancomycin-resistant organisms. So there's really, more research needs to be done on it. I think that's the most we can say right now.
Yes, Dr.
Maybe I can add that the data initially came from the pediatric physicians, but there was a recent publication from a large registry that did not support the use of vancomycin even in children. And honestly, I don't know any convincing data from the adult patient community that would support the use of vancomycin. Clearly, I think that does not mean that the microbiome, intestinal and also biliary microbiome, does not play a huge role in disease pathogenesis, but taking an antibiotic that is not targeting any specific bacterial species probably is not the way to go.
I mean, also in Europe, we don't use it for PSC. There's also a lot of literature in IBD about patients coming into remission, and many of them actually have hidden C. diff infection on top of the IBD, which may explain the efficacy. I think it's an interesting drug. It also has immunoregulatory activity in addition to microbiota. And in terms of microbiota modulation, many of the bugs which were of interest, like Klebsiella in the pathogenesis, for example, are not hit by vancomycin. So I think, of course, it may have multiple also off-target effects, and I think it needs to be studied a little bit more careful. I would say there's not that much interest in Europe as there is in the United States. And I think a lot of the intention, if you look at it objectively, is more single case reports, a very small series, which may come down to precision medicine. The question, perhaps it works in some patients and doesn't work in others. That's something, precision medicine is a big topic in PSC, of course, but it needs to be studied more thoroughly before having a broad recommendation.
Thank you to all our panelists. I also just want to highlight something that Mary entered into the chat, which is that the WIND initiative, which is our natural history database collection initiative that we'll learn more about tomorrow, would capture the use of vancomycin in a more systematic way and will help us understand if there is any kind of association between vancomycin use and PSC progression. But of course, you would need a more a prospective trial, as we call it, to really understand the role that vancomycin might be playing. So we only have about 4 minutes left. This has really flown by. I wanted to ask for our panelists, in thinking about moving forward with this plan. You all have been doing research for many years, and you've been involved in the space for many years. What are some potential pitfalls that you think we should be looking out for? And how can we go about, plan in advance to try to overcome or prevent those pitfalls when implementing this plan? And I will start with Dr. Bowlus for this one.
I think what's been mentioned previously is, focusing in on the priorities. You have a very comprehensive plan. I think if you direct your resources, sort of distribute them across all the areas, you're not going to likely have many successes. So I think you really have to prioritize where you're going to focus your efforts and go for them. Make sure that you're doing something novel, working with groups that have a record of success, and have very specific objectives and goals to be reached, milestones that need to be achieved.
It's very important. Dr. Trauner, Dr. Schramm, Dr. Pai.
Yeah, I'm sure you are well aware, what is going on in other big programs. We are a small community, and the risk is, that initiatives start to compete each other if they're not well harmonized. And there are even some big, private funded research projects, both in the US and also in Europe, which have moved a lot in the PSC field. I think harmonization and communication is everything, and you can take a critical step in, also voicing the patient's perspective, which is really what counts also for regulators. Yeah, I think your role in terms of harmonizing also what is going on at the moment, I think this is also something which is very important.
Yeah, it's certainly a focus for us at PSC Partners. And one point around study design and implementation, it's critical to have these studies be properly powered, meaning we have enough PSC patients participating in the research studies and enough samples. And so that's one area where we can collaborate, harmonize, and work together.
And adding to what has been said, I think you should take care to cover the whole spectrum of disease, because PSC is not a single phenotype. You have the more inflammatory pediatric form, and then you have what we call small duct PSC. No one really knows what it is, I would say, up to large duct PSC in adults. I think you should try to capture all of this, but characterize the phenotype as good as you can.
Great. Thank you so much. And we have just 1 minute left. I would just like to open it to anybody who has any final thoughts, anything we didn't get to that you want to add, share with our community, or anything to comment on related to the ICRNetwork or our agenda.
Thank you so much, Dr. Kirkpatrick, for leading this panel discussion.
Yeah.
Thank you very much. It was great.
Yeah, thank you. I think the comments in the Q&A really reflect how involved the patients are in this process. I think that's great to see. Absolutely. So I will now conclude. Thank all of our panelists once again for dedicating your time in this discussion, and then really dedicating your careers to our rare disease. I'm so appreciative as a family member of a PSC patient. I think that we can all agree that what you do is so important. And so with that, I will conclude the session. Thank you all for being here.
Thank you.
Thank you.